you guys.
Yes.
Hello, everyone, and welcome to today's live broadcast, Improvements to Bile Collection Studies in Rats. I'm Susie Valdez of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by SAI Infusion Technologies. For more information about our sponsor, please visit their site at sai-infusion.com. So let's get started. I want to remind everyone that this event is interactive, and we encourage you to participate by submitting as many questions as you want at any time during the presentation. To do so, simply type them into the Ask a Question box and click Send. If you have any trouble seeing or hearing this presentation, just click on the support tab found at the top right of your presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd now like to introduce our presenter, Merryl Cramer from DMPK and Sampling Specialist at SAI, and Brad Gien, the Global Head of Surgery at Envigo, and Gina, Scientist 1 in vivo pharmacology at Gien Therapeutics. For a complete biography of our presenter, please visit the Biography tab at the top of your screen. Merryl, you may now begin your presentation.
Hello, everyone. Thank you, Susie. As Susie mentioned, I'm Merryl Cramer, and I'm the DMPK and Sampling Specialist for SAI Infusion Technologies. In case you're not familiar with SAI, we specialize in catheters, infusion, and sampling. We're the industry leader in catheter technology and research, including everything from rounded tip catheters for maximum patency to pumps. Today, we're very lucky to have these two extremely qualified professionals on board to help us with different aspects of a successful bile study. Brad is the global head of surgery for Envigo and will be sharing his extensive knowledge of bile duct catheterization, surgery, and recovery. Gina is a scientist at Gien Therapeutics, and she will be sharing some of her firsthand experience running bile studies in the lab. Thank you so much for sharing your expertise, Brad and Gina. Today's webinar will be a top-level overview of bile collection studies with a deep dive into surgery, directed mainly towards people who are new to this model. However, even a seasoned pro will find some useful information in here. It will take about an hour, and we've broken it down into these 6 sections. We'll begin by providing rationale for running a bile study, then move on to discuss best practices when implanting catheters and running a study. Finally, we provide information on the equipment you need to get started and some tips and tricks to help navigate complications and ensure success. So, what is bile? Where do you get it? Why do we need it? If you never thought too much about bile and decided to look it up online, you would find some very interesting information from some very interesting disciplines. For instance, yellow bile is one of the 4 galenic humors developed by Hippocrates. And if you only looked up the definition, you would see that although bile is a luminous gold color, the choleric temperament doesn't have the most glowing reputation. Then again, if you've ever been frustrated by a bile collection study, you might agree that these definitions do kind of encapsulate the bile study experience. Luckily, we're here to tell you how to redefine that experience into one not nearly so angry and gloomy. Speaking of defining, here are the most important definitions to know when running bile studies. I call these the ABCD of bile. Thorough profiling of novel compounds centers on ADME. This is the absorption, distribution, metabolism, and excretion profile of the parent compound, which is what you dosed, and its metabolites, which are generated by metabolism of the parent compound, mainly in the liver. The basics of a bile collection study, or a BDC study, involve surgically implanting purpose-built catheters into both the bile duct and the duodenum. These catheters are externalized and connected to create an external loop which shunts the outgoing bile from the bile duct back down and into the duodenum. This recirculation of bile provides the animal with the bile it needs while recovering from the catheter implantation surgery. This surgical preparation, along with the dosing and collection of bile required, can be intricate and technically complex. Therefore, bile studies are only run when the data they provide is necessary to proceed with compound profiling. The investigation of these pharmaceuticals or agrochemicals, which are in discovery and development, generally involves investigating metabolism and excretion of the compounds in urine and feces. However, it's often necessary to determine the excretion of compound-related material in the bile, especially where enterohepatic recirculation may occur. BDC studies help determine the level of excretion of test compounds into the bile or the effect of the test compound on the physiological system. Overall, BDC studies help us to understand a compound's kinetic profile, in particular the excretion phase, which you may remember puts the E in ADME. And bile studies are frequently required to, as you see on the slide, comply with regulatory guidelines, better understand a compound with unusual PK, to further evaluate specific types of compounds, or to create a stronger base of data for future studies. Okay, we know we need to run these studies, but what information will be gained? As I mentioned, BDC studies inform ADME, which is a regulatory requirement. And right before excretion is metabolism, and most metabolism occurs in the liver. And one of the liver's principal functions is the formation of bile, which is requisite for the digestion of fat and the elimination of drugs and metabolites. Hepatic metabolism is an important determinant of drug clearance and bioavailability. And as we know, rats are frequently the first species used to characterize drug clearance and bioavailability. And because rats lack gallbladders, they are ideal models for examining biliary drug disposition. So this isn't a clinical science talk. So here's just one quick example of a time when running a bile study is well worth the time and the effort. When generic companies are trying to prove bioequivalence between their generic drug and the current drug on the market, they want to demonstrate that their generic drug is highly soluble and highly permeable. This is normally done in a CACO2 cell assay. However, CACO2 cells will not work on compounds which undergo efflux. Luckily, our bile rats can demonstrate that permeability, or lack thereof, readily. So in the case where the generic drug is shown to be a class 1, which is both highly soluble and highly permeable, the generic company can obtain a BCS class 1 waiver and save millions of dollars in development. Of course, this is a great motivation to run a successful bile study. And that is just one example of a time when you would run one. So now we have an example of why we might want to run a bile study, but the question remains, how do we do it and how do we do it successfully? Here are the 6 steps. Look how easy I make it look. 6 simple steps. But those of you who are already familiar with bile collection studies can probably attest to how challenging these steps can actually be. From the surgery to the collection, this is a demanding model. Thankfully, SAI has some suggestions that can revolutionize your bile study experience. And speaking as someone who has run countless bile studies myself, the bile setup from SAI is a real game changer. So here's Brad to tell you about the first 2 steps, which are the surgery and recovery. Please take it away, Brad.
Thank you, Merrill. Good morning, everyone. I am going to talk a little bit about the bile duct surgery process. I have completed thousands of these surgeries, and in Envigo, we've completed tens of thousands of these surgeries. I'm going to move rather quickly through it because I do have a lot of information, and they told me I do not have 4 to 5 hours to present. I have to get through this in about 30 minutes. So we'll move through this fairly quickly. Today we're going to talk about bile duct dual loop surgery, so where we're looping it from the bile duct around the back of the animal back into the duodenum, basically making an extension of the bile duct for sampling. So as part of the surgery technique, we have found it best that rats will be over 250 grams at the time of the surgery. Not to say that we haven't done smaller animals and mice as well, but for rats, we find they thrive best if they're over 250 grams. 275 to 325 grams is optimal for the surgery. Some of the surgery preparation is you want to make sure that you have the correct surgical instruments and supplies. My fancy animation here of all the supplies that we use for bile duct surgery are filling up now. Not going to go through every single thing on the list, you're welcome to review this and pause it and look at what we have on the list. It's very important you have the right instruments when you go to do the surgery. I've seen many times people starting, or I'm training them on surgery without the right instruments, and bile duct surgery is already very difficult, and with the right instruments makes it even that much harder. Part of the surgery prep for us is catheter choice, and this really matters. Starting 20 years ago, I built my own catheters, and I wasn't good at it. I discovered how difficult it is to build a good quality, the correct size, material, and design of the catheter, which is why we went to professionals at SAI to have them custom make our bile duct duodenal catheters. We found that 3 French, which is rather large and slightly more difficult to get into a bile duct, provides the best flow and matches the interior size of a bile duct in a rat the size that we're using. We made it a little easier. We put a bevel on the bile duct end. We're using medical grade polyurethane, so it's not that stiff, but stiff enough that it doesn't just kink every time we put it in. Your other options are polyethylene, which is much too stiff and can damage the bile duct, or silicone, which any of you that have used silicone before knows that it kinks over on itself fairly readily and is much more difficult to work with in these circumstances. We use a bead on the duodenal end and use a purse string suture to put it in. So immediate pre-surgical prep continuing. You want to look at the analgesic dosing, and you want to dose it pre-surgery. We found that to be the best, using inhalation anesthesia to be the best. We have done both injectable and inhalation, and we always pre-dose with analgesic before surgery. We tend to use a buprenorphine, and we've just started using the wonderful extended release buprenorphine from Fidelis, and also meloxicam on board as well. We weigh, shave, and disinfect the skin pre-surgically, and then we use aseptic surgical technique. We complete the surgery in a dedicated surgery area. This can be anywhere that's kind of a low traffic area. We worked in several labs where it's been a table, not necessarily in the lunchroom, but very close to it, to where we have now dedicated surgery buildings. They're very low traffic. We have positive air pressure and airflow, which keeps that area clean. Surgery support technicians are great. Consider them kind of the backbone of the operations there, behind, supporting the surgeon. And having a safe heat source to measure and maintain the animal's body temperature during surgery is also very important. Sterilize the surgical instruments. Without sterile instruments for any surgery, you're going to have infection or complications. And sterilize catheters. And we tend to use a lot of the catheter access buttons for these surgeries. You want to make sure you have a clean area around the surgery in your surgery area, so you're not setting your instruments down in a dirty area or touching dirty areas and then bringing that to the animal. You want to make sure that your surgeons are trained and certified and they have a comfortable chair. Every time I put this in, I have to make sure they have comfortable chair or comfortable shoes if they're standing. Some people, like myself, that are getting old eyes, require a microscope and music, most definitely. So there's 2 methods that I've trained on in surgery. One method we do not use anymore. It's where we make a rather large abdominal incision. We remove the abdominal contents, locate the bile duct and insert the catheter, locate the duodenum and insert the other end of the catheter, tunnel the catheters, and then kind of put all the abdominal contents back into the animal and finish by sewing up everything. Very important during that surgical procedure, you keep the abdominal contents very moist and you're not letting them dry out. much improved procedure, in my opinion, would be more of a keyhole surgery. So we make a very small 1.5 to 2 centimeter incision directly above the bile duct. We use sterile saline-soaked cotton-tipped applicators to gently move the liver out of the way. We locate the bile duct, insert a catheter, locate the duodenum, and insert the other end of the catheter, tunnel the catheters, and then close the abdominal wound. This is much more efficient. This is much preferred for the animal as well, because you're not opening up such a large incision and removing the abdominal contents. And this is going to be the procedure that we go through step by step very quickly, again, because of time constraints. So this is kind of a standard operating surgical procedure for us that we tend to follow and train our surgeons on. So catheterization of the bile duct, we like to go through, having 2 sets of instruments for this surgery, one designated for outside the animal or the skin, and one set of instruments designated for subcutaneous tissue or inside the animal, so that we can keep the aseptic technique intact. So first we would make that incision in the muscle and skin and muscle, and then we would puncture through the muscle wall outside of where the incision is, so as not to have the catheters exiting the muscle through the same incision that we're going to actually complete the insertion of the catheters. This helps the muscle incision heal a lot better and a lot faster, as well as the catheters aren't free and moving. They're actually quite tight within the muscle, so that they can't be pulled out if the animal moves in a certain direction, or if the researcher happens to pull up on the catheter from outside the animal. So we do have fixed cuffs on the catheter. They're placed inside the abdominal cavity, which also helps hold them inside the muscle wall. We try to avoid suturing anything to the muscle wall. It does cause irritation as well. So once we have the catheters in place, we locate the bile duct and gently elevate the bile duct. And if you make your incision correctly, the bile duct is right there. There's no missing it. These incisions in these pictures, again, keep in mind, and the drape being there in one picture and removed in the other, are for demonstration purposes and training purposes. So the incisions are a little bit larger and the drape is missing in some of the pictures, so that we could get a better visualization of the surgery. So you can see in the top right-hand corner a close-up of the elevated bile duct. And keep in mind, the bile duct is not as elastic as it appears in these pictures, or as a jugular vein or carotid would be in a normal vascular surgery. So you have to be very careful not to pull or stretch that duct too much. We place a ligature around the duct and a ligature very near the duodenum, and we place slight tension on the bile duct end of the ligature while we're inserting the catheter. So using micro scissors or a needle, I prefer a small gauge needle, but using micro scissors or a small gauge needle, you can make a small hole into the bile duct, and you implant the bile duct beveled end part of the catheter into the duct. You would tighten your loose ligature on the cranial side of the duct, which will hold in your bile duct catheter. The bile should be observed flowing immediately and easily through the catheter. So once you remove your elevator, you'll see bile flowing freely into the catheter. You do want to make sure that you do cap off that or use a hemostat to crimp the catheter so as to not have bile ducts free flowing out that you still haven't implanted into the duodenum. So once you, excuse me, once you remove and you've blocked off the bile, you've made sure the bile is flowing, blocked it off, you start your catheterization of the duodenum. The catheterization of the duodenum is much easier than finding and catheterizing the bile duct, so this procedure should take you a couple minutes to get done. You want to place your forceps at the tip of the pyloric sphincter, and you want to put in a purse string suture into the duodenum, being mindful to stay away from any vessels that you can visually see. Once you get the purse string suture into the duodenum, right in the center of that purse string suture, you're going to use a clean, sterile needle. We use a 16 or 18 gauge needle, depending on surgeon preference, and you're going to put the needle in, and as you're removing the needle, you're going to be sliding the duodenal catheter at the same time. You're going to discard that needle. It's a single use only, obviously, since we are putting it into the duodenum. And you're going to continue advancing the catheter until you push through the first of two fixed beads. The idea is you're going to have one fixed bead inside the duodenum and one fixed bead on the outside of the duodenum, so that when you tighten that purse string suture, you have a good, clean duodenal area so that no leakage will come from the duodenum. You want to make sure that you draw the duodenum around the sutures and apply 1 to 2 tiny drops of tissue adhesive. I can't, I guess, emphasize enough that you want as little tissue adhesive as possible. I've seen certain animals where it looks like they've used a bottle of tissue adhesive and you have kind of everything stuck together inside. We attach a small gauge blunt tip needle to the end of our tissue glue and place 1 drop of tissue glue just over to make sure that that hole in the duodenum is perfectly sealed before moving on. We do have the 2 beads on there as well that do ensure a good seal. The other option, catheters that I've used before do have a mesh on them. We've had good success with the mesh as well. It's just not preferred for us in this surgery. We did increase adhesion using it, so we moved away from it and stuck with a purse string suture. But the mesh still will work. Once the adhesive is dry, that's kind of key. You got to wait for the adhesive to dry. And in this picture, I will say it does need to be replaced because that is a little much adhesive for my liking. But once the adhesive is dried, then you would release your elevator, which is the sterile saline-soaked cotton tip applicator, and you would flush the catheter with a little bit of sterile water just to ensure that there are no leaks from your purse string suture or glue. You return the duodenum back to its natural position and you exteriorize the catheter. What we like to do is put a little bit of Baytril into the abdominal cavity as an IP injection. And I think I say this on a later slide too. You shouldn't use a whole lot of antibiotic in this case to compensate for poor surgery technique. You want to make sure that your surgery is clean and everything is as aseptic as possible. We do only give a single dose of the antibiotic at this time. Once you trocar the catheters to between the shoulder blades of the animal, we use staples to close the skin incision. We close the muscle incision with sutures, close the skin incision with staples because the animals tend to get at that incision. And then we exteriorize the catheters between the shoulder blades. Standard closing for us is using a stainless steel tube to connect the fixed catheters between the shoulder blades. Obviously, we first make sure that we have bile flowing and that the duodenal end of the catheter is primed. If there's a lot of air in the duodenal end of the catheter, we find the bile will flow through. So you want to make sure that's primed and creating a siphon-like action to get the bile flowing through the catheter that's extending your bile. Here's kind of a finished of a fixed catheter bile duct, which we used to do a lot of, and now we're connecting buttons to these, which Merryl and Gina are going to continue to talk about moving forward, and do a couple of slides showing different exteriorization options. Nothing is more exciting to me and to the surgeons at this point when you have nice, bright yellow bile flowing through your catheter. You can do a number of different exteriorizations. The stainless steel connector, again, is probably, I started in 99% of the surgeries I did early on were that way. We did a few harnesses, and we moved on now, and we've done a lot of buttons. The button is really convenient because internal studies and some external researchers that we've partnered with have been able to keep their bile duct animals around for 30 days or longer, doing studies on Monday, Tuesday, and washing them out, and then redoing some studies in the following week. We kept animals in-house for 30 days, and we did not see much bile duct engorgement. There was a little, but not much. And we only looked at the animals once a week and flushed the duodenal end to stop the buildup of bile salts in that end of the catheter, and took a small sample of bile just to make sure that we still had bile flowing. The buttons and caps allow group housing, which helps in recovery a lot. These animals are very well recovered 5 days post-op, which is very impressive compared to when we started doing these surgeries in our lab many years ago. And the caps also have a handy little window there that you can actually watch your bile flowing if you have good enough eyes. We'll talk a little bit about recovery, welfare, and patency checks. So depending on exteriorization, still housed. If there's exposed catheters, of course, because you do not want the animals that they're sharing a cage with to chew on the bile duct catheters. There's nothing more disappointing than coming in and having a disconnected bile duct catheter or chewed catheter. And paired housings with the catheter access button and the caps, the recirculation caps. This is a preferred method for us, again, because of animal welfare reasons. We do see quite an increase in, or a decrease in recovery time, and increase in better health and welfare of the animals. And we also are able to run more studies contributing to the 3 Rs on the same set of animals rather than doing new surgery on every set of animals. We do supplemental heating post-surgery 24 to 72 hours, along with our sustained release buprenorphine. And we put soft food on the cage floor, which we replace frequently. The animals will step in it, it'll spill, it'll spoil quickly. So we do soft food and gel foods on the floor of the cage in a little dish for them. I don't know about anyone else after surgery. I don't feel like reaching up to grab my food. I'd rather have it close to me. And we do a lot of frequent welfare checks on these animals, being that it is major surgery. Minimally, it's twice a day, but we do check them probably 4 or 5 times a day. During the first 24 to 48 hours, we try our hardest not to take them out of the cage, because a major abdominal surgery, I wouldn't want to be picked up and manipulated. So we try to leave them in their home cage. We do weigh them and make sure to do a full clinical observation panel on them. And if there is a problem, then yes, we will take them out of the cage and either offer them more treatment or warm fluid injections. So we again, try to let them get comfortable in their cages. We observe bile flow, try to observe it every day. Sometimes immediately post-surgery, you're going to see the bile stop. And we've attributed that to kind of the spasm of the bile duct, or the, just the shock of putting in a bile duct catheter. We leave them for a few days, and then we'll look at them again and again in a few animals per thousand, we're going to see positional problems with the catheter. It just hasn't been laid in the abdomen correctly, and it may be kinked, or when the animal rolls up to sleep, it kind of prevents flow. But for the most part, once the animal has had a couple of days to recover, you do see normal bile flow. All right, sorry. So animal welfare. So we switched surgeries from removing everything from the abdomen to just this keyhole surgery because we wanted to reduce internal organ adhesions. In all surgeries, of course, you can have adhesions, but we wanted to minimize it as much as possible. Because the adhesions cause pain, they can lower the catheter patency as well. We wanted to make sure that all surgeons that do this are updated in their training and methods for doing it and have the right equipment and instruments. Less is best. Again, lower amounts of surgical manipulation is a lot better than removing everything from the abdomen. So we started doing keyhole surgery. That's where we saw a great increase in patency. And one thing that early on I didn't think about, we wanted to look at, again, less foreign material left behind. So we cut down our suture tags down lower. It matters. Instead of putting in 5 or 6 different suture points or sutures into the muscle, if it wasn't needed, we don't do it. Powder-free gloves, which I think almost everyone is using or hopefully should be using, especially for surgery, are washing the powder off. We saw some horrible infections long before my time at Envigo with powdered gloves going directly into animals. And just infection control. So you want to make sure that you don't have any bile or duodenal leakage. Anyone that's done this surgery, if you have any bile leakage or duodenal leakage, you know that is kind of a very serious endpoint for that subject. We came up with flushing the abdominal cavity immediately post-surgery with warm saline and kind of flush it and dump it out and flush it again so that we can minimize any leakage that we maybe didn't see at the time of the surgery. Patency, so we can attain several weeks of patency with good surgical technique and replacement. And but again, like I said earlier, bile duct patency can be maintained for long periods of time without any bile duct engorgement and without any ill effects to the animal. What can affect patency? Poor surgical techniques is probably the biggest thing that I saw. Adhesions, which come with and excess suture materials, incorrect or wrong use of surgical instruments, and just surgeon training and practice. It is true what they say, practice, you do get better with practice, or your parents probably told you practice makes perfect. It is true. Catheter placement, so where you place a catheter in the bile duct matters, where you place it in the duodenum matters, and placement, how you lay it in the abdominal cavity. After you go through all that work of getting, your perfect bile flow and great placement in the duodenum, I've seen a lot of people just kind of shove it into the abdomen or subcutaneously when they're powering it, just kind of put it wherever, and they get cancer, they get many other issues. So it's important, even all details on these major surgeries. Catheter size, you want to match bile duct size. It's always easier to put in a very small catheter into the bile duct, and you're going to see bile duct engorgement and flow issues. You want to make sure that you have continuous flow and have as few catheter junctions as possible. The more glued junctions you have, is more points that the catheter could fail or leak. And decrease surgery time. Less time on the table is better surgical outcomes, but you definitely don't want to replace the quality with a 6-minute bile duct surgery. Quality is obviously going to be the most important, but you don't want to have a 2-hour bile duct surgery versus where it should be taking you between 20 and 25 minutes for a bile duct surgery. Now, I think this is the end of me.
Yes, it is. This is where I pick up again. So thank you so much, Brad. That was fantastic. I've done quite a few of these surgeries myself, and I still learned a lot from those slides. So now that everyone knows how to put the catheters in, let's talk about how to safely and efficiently access the bile once the animal has recovered. So earlier I said that surgery is the first step in the bile study, but that's not actually true. Before the surgery, you need to choose the proper catheter. And as Brad mentioned, that catheter choice really matters because any catheterized study is going to hinge on three factors, and that is the catheter, the surgery, and the maintenance. It doesn't matter how good any two of those are. If the other one is no good, you are not going to have a patent animal. That goes for any catheterized study. So when you're choosing your catheter, you should be aware that catheter design can include specialized tips, beads for anchoring, fenestrations, all kinds of things. And these options are all designed to facilitate access to the appropriate vessel. And for bile surgery, SAI has a variety of options, and they're designed to meet the needs of the surgeons who are implanting these catheters. But overall, each catheter is going to have a very small tip in order to access the very small and friable bile duct. As Brad explained, we recommend a medical grade polyurethane catheter because polyethylene is a bit too stiff and silicone is a bit too flexible. So medical grade polyurethane is definitely the way to go. And of course, we can provide a 3 French medical grade PU catheter that has a beveled tip on the bile end and a bead on the duodenal end, or we can customize the catheter further for what your surgeon requires. For the duodenal catheter, you can either implant using the purse string suture as Brad described, or you could select a catheter with mesh on the end so that you can secure it in place with a tiny amount of vet bond or another sterile tissue glue. But once you've selected the proper catheter for your surgery and your catheters are placed and externalized properly, the way Brad explained, you have a few options when it comes to connecting the bile circuit externally. And here is an illustration of 3 of them. So the first one is you simply externalize the bile duct and duodenal catheters and connect them with a stainless steel connector until you're ready to go. Brad showed a picture of that. And as he mentioned, that's how many surgical vendors complete their bile loops before shipping their animals. They used to always do that, but times have changed. Some people still get their animals this way because they're collecting bile intermittently while the animal is anesthetized, or if they're using an autosampler such as the BASI QLEX. The issue here with this connection is that you need to manipulate a very small and very slippery connector or pin, as well as the catheter, which is continually leaking slippery, soapy bile. And because of this, pins can easily slip out of the catheter, leading to lost samples and potentially lost animals, which is awful because obviously you don't want to lose any animals, but also now you're down an animal and you spent a lot of money and time, and you want to avoid losing catheter connection at all costs. The next option, the second picture, is the harness. And if you receive your animals with the externalized catheters that I just described from the first picture, you can apply this harness to your animal in-house. Vendors are happy to put it on for you before they ship the animals if you want, and that may be preferable since attaching the harness requires connecting those same slippery catheters to the pins on the underside of the harness. But once applied, the connection is protected by the dome of the harness, and the catheters can be accessed through the ports on the harness. These harnesses are available in 1, 2, or 3 ports, and they're affordable. A short tether is used to recirculate the bile, and while on study, these animals can be hooked up to a quick connect tether for a secure long-term bile collection. However, you need to be diligent about adjusting the tension of the harness to ensure a proper fit. As the animal moves, grows, loses weight, you need to adjust the tension on the belly bands to make sure that the harness is fitting properly. Finally, the third option is the button that we've been discussing. Your surgical vendor can implant that cannula button during the catheterization surgery. And with this button, up to 3 catheters can be securely attached to the unique barbed cannula pins, which are underneath the skin. These connections are well protected and extremely secure. To recirculate the bile, you simply place that shunting cap that you saw earlier on the button. And this cap pops right on and attaches magnetically for an extremely easy, quick, and secure recirculation of the bile. The rats can then co-house while recovering from surgery, and that, of course, is a major refinement for animal welfare. And then when you're ready to begin your study, you just remove the cap, and the tether connects just as quickly and easily. You just pop it on, and you can keep the animal tethered with no worries about discomfort or fit. And since the cannula button is by far the fastest and easiest method and has extremely high success rates, I'm going to focus on it for the rest of this webinar. Here again is the shunting cap, and you saw it in Brad's slide earlier, and this is just a very quick and simple way to recirculate the bile. The cap contains a U-shaped tube which connects the bile line to the duodenal line. And as I said, it connects magnetically to the implanted button and pierces each closed septum of the bile button, which allows the bile to flow up and over and back down into the animal. And the route is very short. It's a very short piece of tubing, and the head space pressure is very low, so the bile has an easy time flowing while the animal recovers from surgery. And then because of the closed septum on the button, you can remove the cap and replace the cap without any worry about the bile flowing all over the place when you disconnect. Bile is a slippery and unpleasant substance, and keeping your animal clean is so much easier with this shunting cap. Not to mention, connecting, disconnecting, and reconnecting can all be done in just a couple of seconds. While you're waiting for your rats to recover from surgery, you can set up your cage system. Bile studies are normally run in a metabolic cage or a metabolism cage in order to collect the rest of the excreted compound, which is found in the urine and feces. To adapt a metabolism cage lid for infusion and or bile collection, you simply drill two holes for the mounting plate and screw the cage mount in place with the 2 screws that are provided. You can see here at the bottom of that aluminum pole, there's a little mount with 2 screws. It's super easy to connect to your cage lid. The tether is then fed up through the lid and attached to the swivel, and the swivel is in the swivel mount. In this picture, the swivel is orange. And you can now attach an extension line to the top of the swivel and run that line down to your collection vessel. And you need to always make sure that your extension line is extending to about 8 inches below your rat. It's critical for bile collection, and I'll talk about that again. So the first setup for a bile study is you're just collecting bile. And if you're only collecting bile, you'll need the bile and duodenal catheters to connect to the 2 pins on a dual button with a shunting cap for recirculation. A single tether connects to a single channel swivel, and then you need the cage mount and extension line, of course. This is the easiest bile study since the setup is so simple. However, you can only collect for about 24 hours max before you need to reattach the shunting cap and allow the rat to recover. Many labs will not collect for over 8 hours without providing replacement salts or artificial bile via infusion into the duodenal catheter. The next option is bile and replacement salts, which is what I just mentioned. If you're infusing artificial bile, you still need the bile and duodenal catheters to connect to 2 cannula pins on a dual button with a shunting cap for recirculation. However, when you start the study, you need a dual bile tether connected to a dual channel swivel to allow free rotation of the animal throughout the study. This time you need 2 extension lines, one for the bile collection and one for the artificial bile infusion. When infusing your artificial bile, labs routinely keep a rat on study for up to 96 hours. I used to do this all the time, but you need to be infusing artificial bile. A study this long comes with comes with its own challenges, but with the K-lock button, there's no issues with the catheters or tethers detaching, and there's no problem with harness fit. You can also collect blood while collecting bile and infusing artificial bile. You simply attach all 3 catheters to a triple button and use a triple tether. You then have access to the bile and duodenal catheters as described above, but you also have access to the venous line as described in the next slide. While you can have a bile duodenum blood setup, this setup is more common. When only collecting bile and blood, you'll need the bile duodenal and vascular catheters to connect to all 3 pins on a triple button. As usual, you'll use a shunting cap for recirculation. And a dual tether connected to a single channel swivel will allow the bile to travel through the swivel while the vascular line hangs out of the tether. You can see it here, hanging out with a handy red catheter access port for clean and easy access when sampling. That line that's hanging out the side with the red port is your vascular line for taking samples. And you can't see it here, but at the top of the yellow swivel, there is an extension line that is collecting the bile. You still need the cage mount and the extension line, of course, but this setup is almost as simple as bile collection only. And once again, you can only collect for about 24 hours if you're not infusing artificial bile salts. So now you know why you're doing it, and you know what you need to make it happen. You just need to know how to do it. So your rat is catheterized and it's been acclimated to the fully connected metabolism cage setup. When you're ready to begin your collection, you simply remove the shunting cap and replace it with the appropriate tether. You always ensure that the end of the extension line is well below the rat in order to create the essential siphon action, which pulls the bile up and over the headspace of the tether and swivel mount. I can't stress this enough. Once you take the shunting cap off, keep the end of the line below the rat. Some people like to fill the tether and extension line with fluid in order to encourage this siphon action, but I never did that. I was always worried about dilution of the bile in the tether, but different labs have different methods, and all labs need to ensure that the siphon action is never impeded. If at all possible, hold the connected rat above the system with the tether angled down until the bile has traveled well down into the extension line so that when the system is placed upright again, the siphon action is maintained. When you're getting a bile study going, the siphon action which pulls the bile through the catheter is 90% of your problems. You're going to want to do everything you can to maintain that siphon action. If you have a PO dose, you'll want someone to hold the lid and tether while you hold the rat for dosing. Because it's all hooked up to the cage. If you're IV dosing, you can leave the rat in the cage and dose through the catheter without it. In patency, just send me an email. I'm more than happy to share our previous webinar, which covers these topics in detail, or we can email, talk on the phone, whatever. I love talking about this stuff. So once your animal is dosed, don't forget that the bile still has to travel to the collection vessel. You're going to have some untreated bile in your first collection fraction because you dose them, and then the bile that the animal has been dosed, the bile has to move all the way from the animal into the collection vessel. So you're going to have some blank bile in there with your treated bile. So bile generally flows up to usually 1.5 mils per hour. So you're going to want to make sure you have the appropriate collection tube for the period of time you're collecting. You don't want to have too small of a tube and overflow. That would be disastrous. Unlike blood, bile doesn't need to be anticoagulated, and you can keep it on dry ice if that's necessary for stability of your metabolites. And don't forget, once again, you've got to keep that collection tube below the level of the rat, ideally somewhere around 8 inches under the rat. Troubleshooting. This is the most perfect picture ever, and I love it so much. So let's talk about things that can go wrong when we're running a bile study, because if you've ever run one, you know that things can go wrong. Suboptimal bile flow is almost certainly due to catheter placement. An improperly placed catheter can also lead to bile leakage, catheter dislodgement, and stomach bloat. Placing the catheter the other issues you might face with a bile catheter include creation of a false gallbladder, which is when the bile simply pools instead of flowing out of the body. A bile catheter
Merrill,
should never be flushed.
I'm sorry, Merrill, can you hear me? Hi. We lost you a little bit there, so could you start over on this slide again? For some reason, it's dropping here and there.
All right.
Okay. Thanks. Or not. We might have lost her again. Okay. I'm going to take a pause here for a second and see, is everybody able to hear me okay? I'm asking my colleagues here on the line. I can see that they can. So until Merrill, thanks. Until Merrill can jump back on, in which case I'll hear her voice and I'll jump out. I'm going to try and keep this going for us. So so as Merrill was saying, the first round of issues are catheter-related issues. And so this has to do with how the catheter is placed and whether it stays in the right place. And there you are. Hi.
That's it. Can you hear me now?
Yes, It's a little bit choppy. It's a little choppy, but let's try it again. I'll just jump in if I need to where I can.
Well, thanks for telling me. I don't know what's going on. It looks like I've got good reception here, but 2020, am I right? So anyway.
That's right.
So suboptimal bile flow is almost certainly due to catheter placement. I don't know if you heard me say that, but I'm saying it again. An improperly placed catheter can also lead to bile leakage, catheter dislodgement, and stomach bloat. Placing the catheter properly takes quite a bit of practice. Like Brad said, practice is going to help you be almost perfect. And they've done thousands of practice themselves. So I highly recommend purchasing pre-catheterized animals if at all possible. Some of the other issues you might face with a bile catheter include creation of a false gallbladder, which is when bile simply pools instead of flowing out of the body. A bile catheter should never be flushed. It's not necessary, and it will almost definitely cause damage to the bile duct, so never flush the bile catheter. You can flush the duodenal catheter if need be. Additionally, use of an inappropriate catheter can cause issues like discomfort and damage to the bile duct, which is quite friable. And remember, SAI leads the industry in catheters, and we're happy to help you ensure that you have the best catheter for your bile and duodenal surgeries. So if you're doing your own surgery, just ask us for help with the catheter, and we'll make sure you get the right one for your studies. So, the dreaded loss of flow.
Or the dreaded loss of sound again. Great. Okay. I'll try again here, although this one is one that Merrill knows more about. So some of the things that you can do if you lose flow in the system are forcing the rat to change position. So this might mean standing them up, or sometimes a gentle palpation of the abdomen with your fingers can help get things moving. Another thing that you can try, if it doesn't interfere with your protocol, is a treat. That can kind of help things get going in the digestive system, which is what encourages that bile to flow. And then if you are absolutely, totally, completely desperate, you can very, very, very slightly pull on the syringe with a line to motivate the bile to flow. All right.
I don't know if I'm on
there.
or not.
You are.
Am I
Go
here?
for it. Yes.
Okay. I'm not trying to make things difficult. I don't know what's going on. So yeah, you might want to move the rat around a bit to get that flow going. Sometimes the animals are super flexible and they just position themselves in such a way that the catheter is pinched. You make them move around. You can also try gently blowing into the cage. They like to smell your breath. I don't know if it makes them hungry or if it makes them nervous, but it makes their bile flow sometimes, so it can speed up your flow. Of course, if you are allowed to offer them a treat, that can also get their bile pumping. And as Candace mentioned, once you've exhausted every possible option, you can, if you simply can't resist it, use a syringe to pull the very slightest vacuum on that line. And you'll want to hold the slight vacuum for a tiny bit and look and see if the bile is slowly creeping up and over the headspace. And then you want to ever so gently and slowly release that vacuum by removing the syringe. This is a tricky technique, and it can have dire consequences if you do it wrong. So only do that as a last resort. And as far as speed of flow, I believe I've mentioned the siphon action quite a few times. You really need to make sure you maintain siphon action. If your rats are in metabolism cages, you could keep the vials on ice on the bench or even tape them to the leg of the cage stand. You just need to make sure the end of the extension line is below the rat at all times. If you're noticing that the bile is getting dark, this can be a sign of stasis where the bile is simply pooling. In other cases, you may notice bright yellow urine and plasma, and this may be bilirubinuria from a damaged liver, which leads to bilirubin in the bile and the blood. When this happens, there is usually an elevated amount of pest compound in the urine and blood, so you need to bring that to the bioanalyst's attention so they can make a note of it on the results. And now it's time for a case study from Gina. I know we've spent the last few slides discussing problems and potential issues, but I hope I was able to shed some light on solutions to those issues, and I hope that I didn't cut out too much and make it difficult. But speaking of solutions to issues, it's now time for Gina to let us know about her experience running those bile studies. So Gina, what you got?
Thanks, Merrill. So we are kind of running out of time, so I'll try to be brief. This is obviously not me in the picture, but this is a colleague of mine. We still work together to this day, but this is just kind of an example of some of the pain that we experienced when we first started doing bile studies. The buttons were not available at that time, so we did use the harness, and you can kind of see the rat there is anesthetized in the harness. So we got all of our surgerized animals from a vendor. We did not have the vendor put the harness on, so when the rat arrived, we had to single house them. To put the harness on, you do, unless you're really, really good, you have to anesthetize the rat to get that harness on, to disconnect your loop, your duodenal and your bile loop, hook it up to the harness. As Merrill said, it's very slippery. It's quite challenging to get the small cannula onto the pin of the harness, make sure it stays on there. We also had a JVC line because we would infuse IV, our drug IV, to collect bile, and then we would also sample blood as well. So hooking all of that up took a little bit of time, and then we would just leave the rat anesthetized. As you can see, that line is well below the rat. My colleague Kristen there is happily waiting for the bile to flow, which, as Merrill also mentioned, can be quite challenging. So I will just say, again, because we're running short on time, switching to the button has been a lifesaver. It's the ease of use. Anyone can really disconnect the shunt cap, put the other tether on, and as long as you keep that line below the rat, bile will start to flow. Everything is much, much easier. I will say also, from a welfare perspective, we've used the buttons on both rats and mice, and we've also used the harnesses. If you've ever put a harness on a cat, I have a lot of cats, they kind of go flat and they freeze and they're miserable. And if you've ever seen a mouse or a rat in a harness, they're also very miserable because it's going around their belly, it's going around their neck, it's rubbing on their neck and under their shoulders. It's very uncomfortable. I will say the animals connected with the button to the tether line, there is nothing going around their neck or belly. They look, at least, anecdotally, this is just my own personal opinion, they move about the cage more freely, they're less stressed. With that, if the rat is moving more, you're going to get better bile flow. You get usually less positional occlusions. You don't have to, go in there, and I've massaged many an abdomen to try to get bile to flow. The other thing with the harness is your catheter can slip off that pin, and if you don't catch it in time, then bile is just flowing all over the rat, and you come back to switch out your vials for your collections, and you find a big yellow mess in the cage, and the rat's just kind of swimming around there in its own bile in the fur. So anyway, I'll just end by saying ease of use for not only the person doing the study, so again, it doesn't have to be the same person every time, because the quick disconnect kind of, I don't want to say it's idiot-proof, but it is. You take off the shunting cap, you put the tether on, you wait for bile to flow, and then again, the stress to the animal is much less. We only ran our bile studies for 24 hours, so ours were always terminal at the 24-hour time point. But as they mentioned, you can recap the animal, put the shunting cap back on, group house your animals, give them a washout period, replace bile salts, and reuse them. So I'll end with that. Thanks. Sorry I went over. And then I guess we'll probably do questions if there's time.
Thank you so much, Merrill, Brad, Gina, for that informative presentation. And we will now start the live Q&A portion of the webinar. We have time for a few questions to be answered right now, but we want to urge our audience members to go ahead and ask that question by putting it into the Ask a Question box located on the far left of your screen. We'll answer a couple now, and any questions not answered right now will be answered via email. So let's go ahead and take a look, because we have quite a few questions already coming in, and this has been such a great presentation today. Our first question is, how do you decide if you should replace bile salts or not? And I think maybe Merrill or Gina could probably answer this question.
I'll take it. So when you're deciding whether or not you need to replace bile salts, if you need to collect bile, if your exposure curve is longer than 24 hours, meaning that you're still seeing compound in the animal after 24 hours, you're going to want to keep collecting, in which case you really need to be infusing artificial bile. Some people just do a bolus of artificial bile on a schedule. Some people run a pump and infuse it in, but you need to do something. So really, if you're going to do it between 8 and 24 hours, that's just a judgment call by your PI and your vet. They need to decide like at what stage is this study, and is it more screening, and are they able to do the infusion? It's obviously it would be nice if every rat got infused starting at 0, but it's not always possible. So it's just a matter of what you need according to your PI.
Thank you so much. And we have time for one more question. This question is for you, Gina. Does using the counter lock button make it easier to train staff or rotate them?
Definitely. I mean, you still want folks who have an understanding of the bile study. You don't want to just throw anyone in there, because even with the button, there can be challenges, as Merrill said, just waiting for the bile to flow. So once it's connected, again, making sure you keep that line below the rat. It will take a little bit of time, and it can get frustrating. But again, the ease of just not having to have 2 people or 3 people in there kind of anesthetizing rats and holding rats above your head waiting for bile to flow does make it easier to train staff and swap them in and out. Again, making sure, though, that you want to go through those kind of troubleshooting things that can still happen with the button. But again, in terms of ease of use, yes, anyone can connect it.
Thank you so much. And actually, let's get in there and ask one more question, because I want to make sure we get and include Brad in here. Brad, can the counter lock buttons be implanted in mice?
Hi, yeah, thanks for the question. That is That's a great question, and yes, they can. So we have seen and actually changed our standard surgical exteriorization in mice to the cannula buttons. Mice are acrobats. They can kind of twist their head right around and tend to get at catheters a lot. So we have started putting as a standard cannula buttons in all of our mice. And we've seen such an improvement in patency lasting out to, 30 days plus, as well as keeping the catheter safe and clean. So yeah, they work amazing in mice as well.
I want to take the time to thank all three of you, Merrill, Brad, Gina. What a great presentation, and I want to thank you for your important research. I'd also like to thank LabRoots and our sponsor, SAI Infusion Technologies, for underwriting today's educational webcast. Before we go, I want to thank our audience for joining us today and for their interesting questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And you can view this webinar on demand, so LabRoots will alert you via email when it's available for replay. That's all for now. Take care. Until next time, stay safe and stay healthy. Bye-bye now.